going to study about the structure of fermenter. Now fermenter is a giant steel container. Its capacity is 100,000 to 150,000 liters. Now its structure is made up of a cooling jacket. Now what's the function of this cooling jacket? Its function is to ensure the correct temperature. Why the correct temperature should be maintained? Because within the fermenter, the enzymatic activity or the chemical activity is going on, due to which heat is released. Now, when the heat is released, then it causes the enzyme to get denatured. So, to prevent the enzyme from being denatured, the heat which is produced due to the chemical activity is moved out due to this cooling jacket. From one side, the cool water will gain entry, whereas from the other side, the hot water, it will move out so that it could carry away the heat. Clear? Now, sterile air is blown inside the fermenter. Now, why the sterile air is blown? Sterile air is blown so that the air is free of pathogen. It cannot cause any contamination. And it could prevent any unwanted changes so that any other bacteria cannot gain entry. Air, why we supply this fermenter with the air? To provide oxygen for aerobic respiration so that energy is released, and this energy can be used for the cell division of the organism which is growing inside the fermenter, so that the growth of the organism can occur. Now, this sterile air is blown through a metallic plate containing holes in it, which is known as sparger. Now, why it contains holes in it? So that it increases the surface area. For example, if there is no metallic plate, then the oxygen will be present only in this area. Because of these fine holes, the oxygen will be distributed evenly in the whole culture solution, so that it will increase the rate of respiration. Clear? Now this impeller, this impeller is controlled by the motor. Now what's the function of this impeller? Is to mix the content well, is to mix the culture solution with the oxygen. Sometimes as in SCP production, because in SCP production, that is single cell protein production, we grow fungus inside the fermenter, and in order to maintain the filamentous texture of the hyphae of the fungus, the impeller is not used in SCP production. Fine. Why impeller is not used? Because when the impeller rotates, then it could break down the filamentous hyphae. That's why we are not using impeller in case of SCP production. Clear? Now, we will fill this fermenter with the culture solution. The culture solution should provide the carbohydrate source, which could be sucrose or lactose. We should provide the culture solution with the protein source. Protein source could be mineralines. Now, why carbohydrate source should be provided? So that it could provide energy for the growth. Why protein source? So that protein is essential for the growth of the organism. If there isn't any protein, then there won't be any growth. Then the temperature should be maintained from 26 to 30 degrees Celsius. Any temperature you can write down. Fine. Now, pH should be maintained from 5 to 6. Fine. And then when the culture in this this fermenter is filled with the culture solution, when we grow the culture inside the fermenter, then finally this culture solution can be harvested through this outlet. Clear? So this is all about the fermenter. That's it.